Okay, hello and thank you all for attending this webinar today. This is CIL's first joint webinar with MRM Proteomics. It is entitled Stable Isotope-Labeled Standard Peptides for Precise, Robust, and Sensitive Quantitation of Multiple Proteins in Biospecimens using PeptiQuant LC-MRM MS Assay Kits. My name is Christoph Borchers and I'm the Marketing Manager here at Cambridge Isotope Labs. Before we start, I just have a few housekeeping notes that I wanted to note before I introduce the speaker. First, this webinar is being recorded and will be available for electronic viewing after the event. We'll send a follow-up email to all of you and also have it available on our website, isotope.com. Next, if you have any questions during the webinar, please type them into the comments section of the GoToMeeting screen. We will try to get to as many of these questions as possible during the Q&A session following the presentation. Lastly, we ask that you keep your phones on mute during the presentation. This will reduce any background noise, and we thank you in advance. Now it is my pleasure to introduce our speaker, Dr. Christoph Borchers. Dr. Borchers received his bachelor's, master's, and PhD University of Konstanz, Germany. After his postdoc training and employment as a staff scientist at NIEHS in North Carolina, he became the director of the UNC Duke Proteomics facility and held a faculty position at the UNC Medical School in Chapel Hill up until 2006. Since then, Dr. Borchers has been employed at the University of Victoria in Canada and holds the current positions of professor of Biochem and Microbiology and the Don and Eleanor Ricks British Columbia Leadership Chair in Biomedical and Environmental Proteomics. He is also the director of the University of Victoria Genome British Columbia Proteomics Center, which is a member of the Genome Canada-funded Genomics Innovation Network. Dr. Borchers is also appointed as a professor at McGill University in the Department of Oncology in Montreal. There, he received the Siegel Chair in Molecular Oncology at the Jewish General Hospital. His research is centered around the improvement, development, and application of proteomics technologies with a major focus on techniques for quantitative targeted proteomics for clinical diagnostics. Multiplexed LC-MRM MS approaches in immunomulti techniques are of particular interest. Another focus in his research is on technology development and application of the combined approach of protein chemistry and mass spec for structural proteomics. Dr. Borchers has published over 200 peer-reviewed papers in scientific journals and is the founder and CSO of two companies, Creative Molecules and MRM Proteomics. The webinar today will focus on the applications of the MRM PeptiQuant assay kits. CIL is the exclusive worldwide distributor for these kits, so please feel free to contact us with any questions you have about the products or to place an order. Now, without any further delay, it is my pleasure to introduce Dr. Christoph Borchers.
Thank you, Krista. I hope you all can hear me well. If not, please let us know. I also see that some of you that haven't muted the line, please do so. Otherwise, you have some background noise. Again, Krista, thank you for the introduction and thank you for giving me the opportunity to talk about our work, what we're doing in targeted proteomics. I cannot move the slides. Doesn't work. Okay, good. So we're doing this since quite a while, actually, since I came from UNC in 2006 to Victoria, and we and others, then we believe that we can quantify proteins in a very targeted way using technologies including multi-reaction monitoring, and we were quite happy when in end of 2012, Nature Method called it as the method of the year. And in the next slide, I would like to explain to you in a very simple way what multi-reaction monitoring is, and I apologize if it's really too simple. as you know, we quantify proteins by actually analyzing the peptides, which we getting out of the proteins by proteolysis. In most of the cases, we're using trypsin as our enzyme. And since we are analyzing very complex mixtures, including blood, urine, and so on, we have been making out of the thousands of proteins even more peptides. And we need to pre-fractionate this, and we do this by liquid chromatography, which is an online coupled to a mass spectrometry. And in most of the cases, we're using a triple quadrupole, but we also know that we can use some other machines to do this, including the Q Executive and the QTOF instrument. But this shows like how it works with the with the triple quadrupole. So the peptides coming out of the column into the mass spectrometer, it's not one by one, they're coming into the bulk and into the first quadrupole, and the first quadrupole acts like a mass filter, and we select, in this case, this peptide. We're setting the mass. Since it is a target approach, you know exactly what we are looking at a certain time. And then this goes into the collision cell, hits some gas molecule, falls apart, and again, then from there into the third quadrupole. And here we have a second mass filter where we're only looking at a certain kind of fragment, which we also know exactly what is coming out. And in the end, what we're getting is an ion chromatogram, as shown over here. This is nothing really new, has been used in the pharmaceutical companies to quantify drugs and biospecifics. And now we are able to do this with peptides. The advantage is certainly that we have like a very broad linear range, and this is really one of the most sensitive mass spectrometric approach what we know about. But for us, what is very important is, and I hope I get this message across through the seminar, is that we're using internal standards. And these internal standards, they have the same sequence as of the peptide, of the endogenous peptide. However, they are isotopically labeled. It means like and if we're using C13 and 15 as our isotopes, the behavior of these peptides are very much identical, so the endogenous and the standard peptide. However, the standard peptide is a little bit higher in mass, and which we can use then in mass spectrometry, since mass spectrometry is separating molecules based on the mass. So if we're using these cis peptide, then we can approach absolute specificity. We never can say if you have absolute specificity, but at least we can approach it. Since we are looking now for 4 molecular specific criteria, which are the precursor ion, M over Z, so the mass of the ion, and the precursor, the mass of the product ions, and the retention time. The retention time needs to be exactly the same between the cis and the net. And also the response from the cis and net from the fragment ions should be identical. So we have at least like 4 molecular specific criteria we can look at to determine the specificity. The next advantage is that we can quantify our proteins in a very precise way, and sometimes even in an absolute way, but I'll come to this later. What is important here is that we rigorously characterize our cis peptides, and we know exactly the amount and purity of the peptide standard, that we can, even if the standard is running out, we can repeat it and be achieving the same result. And we also, another big advantage is that we're using the cis peptide to tune our instrument. And this leads to fantastic sensitivities. In the end, we can achieve atom-mode sensitivity on column. Another very important advantage is the reproducibility. And most of the cases, we have CVs which are better than 20%. And having these good reproducibility was really the breakthrough in proteomics. On the next slide, you see that we have here, it's a summary of an experiment where we have analyzed 45 plasma protein targets 12 times, and you see the distribution of the CV where we're not normalizing this to anything. Like we're having around, this is pretty much what you know from spectral counting and these label-free approaches. We have a CV between 25 and 30% on average. But if we're using internal standards and normalizing this to internal standards, then we improving the reproducibility, and now we are at 6%. However, we can do it even like in a better way. Like here in this case, we just add the internal standard at the same amount for all of the 45 plasma protein targets. If we do it in a more sophisticated way, what I mean with that is that we spike in the internal standard in that way that we have a 1-to-1 ratio between the endogenous and the cis peptide. That means, like, for example, albumin, we need to, which is present at milligrams per ml, we need to add much more compared to a protein which has a concentration of nanograms per ml. And then if you do this, then we achieving reproducibilities with a CV on average 2 to 4%, and all of them in our case are below 50%, and this is exactly what FDI would like to see. And we also, this on the next slide, we using these cis peptides for interference screening. First of all, it helps us really to improve the specificity, since the cis and the net peptides have the same retention time, and also if you look at the peaks for the net and the cis, they should be very similar and symmetrically. And then we can use the ratios of these ions to determine interference. When we're looking at the ratio, for example, in the cis, we can spike it in buffer, and we're looking at the ratio, in this case, from the Y5 to Y4, which is 0.22, and this ratio should be identical of the cis in the matrix. When we spike it in the matrix, in this case, plasma, and we also have this, like the cis is like Y5 to Y4 is 0.22, and the net should have the same ratio, which is shown over here, Y5 to Y4 is 0.22. And if you see this, then we can be quite sure that we don't have any interference. So this is a great tool to check for interferences. Having internal standards, and it's quite important, having very precise quantitation is quite important, particularly for biomarker validation. And we came all together, like I think it was like last year in Bethesda, and we discussed how can we make this proteomics or the whole society more I would say, like, what's the right word for this? Like, make sure that we're doing the good job on this, that we're really reproducible. And particularly when it comes like to biomarker validation, we need to be precise, and which is shown as tier number 2. Tier number 1 is really like something for the clinic, where we not only have precision, we also need to have accuracy. But in most of the cases, we don't really need this, but we certainly need to be precise, and this is what we can achieve when we're using internal standards. So on the next slide, I have listed like what needs to be done when we develop an MRM-based assay. Since it is a target approach, we need to know what we're looking for. So most of the clients that we have, they're coming with a list of proteins which I would like to have quantified. And from there, we need to select the right peptide, which is sometimes not that easy. But I come to this a little bit later. When we have the peptide, then we synthesize the standard peptide, and we're characterizing the peptide quite rigorously through AAA and CZE. And then we using the peptide, we're optimizing our instrumentation, so the LC and the mass spectrometer, and then from there we building up the assay and validate the assay. And means like we're looking for repeatability, reproducibility, accuracy, linear range, and so on, like all the other things like lower and upper limit of quantitation. And then we are ready to go and can analyze our samples. So I mentioned before that selecting the right peptide is really the tricky part, and for that we have our own criteria as what we're looking. looking at and what needs to be fulfilled. First of all, the peptide needs to be unique to the protein. Then the peptide shouldn't be too short and not too long, and it shouldn't have any miscleavage site and other criteria, like we try to avoid easy modifiable amino acid residues like cysteine, methionine, and we're also looking at the sequence in great detail, that we don't have sequential choline and ascholine. We're trying to avoid it as well. Though the means like we not only looking at the sequence by itself, we're also looking at proteomics databases hasn't been detected before, and then we're looking at protein databases. Are there in this peptides potential post-translation modifications in there? And then also we're looking into genomics databases. Are there some SNPs in this peptide being reported, and so on and so on. This is a pretty time-consuming step, and though that was the reason like why we wrote this program, which is called Peptide Picker, and this actually it's accessible online and everyone can use it, and that really speeds this up, the selection of the protein. It takes around 1.5 minutes. Normally it takes like really hours if you do it manually going through it. And then you have a list of peptides which you think are suitable, and we give them a score, and from there then we synthesize the peptides. We're using FMOC chemistry for synthesizing the peptide, and we have it isotope-labeled, and since we are using trypsin as our enzyme, at the C-terminus we have either a lysine or an arginine residue, and we start from there. When we're doing our FMOC goes from the C to the N-terminus of the protein, we start from the isotopic-labeled lysine and arginine residue. And after that, we purify the peptide by reverse spec HPLC. There's an MS detector on it that is just selected the fraction of our interest, and then we're characterizing the peptide rigorously by molar topomers, looking for impurities, amino acid analysis, and calculated zone net-free that we finally have exactly the amount and then the purity. So from there, we have actually built up like quite a library of cis peptide. We put it in our peptide tracker, and which is also like available for the community. You can look what we have so far in our peptide tracker. So far we have around 900 peptides for around, I think total 2,500 peptides that we have, which corresponds to 900 human proteins, 800 mouse proteins, 500 rat proteins, 300 pig proteins, and so on. And everything is pretty much listed in this peptide tracker, and you can view it, what is there, and you see like we have of course like the species, the protein name, and very important the Unicode number, and then we have the peptide which you are using for quantifying the protein, and then we have all the transitions and the dwell time, all the LCMS parameters that are accessible and can be used. So, and then when we have our peptides, and when we have developed our assay using the peptides, then we are ready to go. And here is a general method overview for plasma analysis. First, we need to denaturate the plasma. There are different approaches how to do this. But important is like you need to write the protocol for one approach and really stick to it, and not switching the protocol. It's totally different. You get different results if you're using, for example, urea versus deoxycholide for most of this protein. Then we reduce, alkylate, quench, digest, and so on. We're adding the peptides in a balanced way, as I mentioned before, and then we do a quick cleanup, and then it goes from there into our standard flow UPLC MMS. This entire approach to sample preparation can be done in a fully automated way. We have 2 liquid handling systems in our laboratory. There's a TCAN, and now we're moving more over to the Assay Biomarker because it has a 96 channel has, so we can do 96 at the same time. And with the TCAN right now, we only can do 8. And then we determine the standard curve by adding the cis peptide a different amount, as you can see over here. The net is constant. And then we get the standard curve, and ideally then we spike in here this amount of cis peptide to our samples we want to analyze, that we have around a 1-to-1 ratio. And from there we run then the samples, and we getting our output out. So here are a few examples of what we have developed so far, what assays we have developed. I start with plasma. Our highly multiplexed assay is able to quantify around 190 proteins in human plasma, which corresponds to 483 interference-free peptides. And this assay spans almost like 6 orders of magnitude in concentration range in which the proteins are present. That means that we are able to quantify serum albumin and at a concentration of around 30 milligrams per ml down to myeloplastine at around 20 nanograms per ml. And in this assay, there are many putative biomarkers already in there. According to the literature, we have 81 CBD biomarkers, and we have 65 cancer biomarkers. There's a lot of apolipoproteins, which is of great interest for many researchers. So, not only doing it only for human, also for mouse. As you can see over here, in this assay, we can quantify around 100 mouse plasma proteins. And again, like we're covering 6 orders of magnitude. But if you want to go deeper into the proteomes, and like that's just jumping back, it's like here, this is like around like 50 nanograms per ml. If you want to go into ideally into the sub-nanograms per ml or very low nanograms per ml, then we need to pre-fractionate the plasma prior to our LC-MRM analysis. And we do it with high pH HPLC, and you put it into 96 circlets, and we pool these fractions together in that way that we have on average around like 50 proteins per run. In this case, we have 13. And then we have a pretty equal distribution of chromatographic resolution. And so now we are able actually not only spend 6 orders of magnitude, it means like 100 milligrams per ml, so around 30-40 to around 30-40 nanograms per ml. Now we can even go deeper, as you can see in the green rectangle. And we only can achieve this when we pre-fractionate this. Down to the low, very low nanograms per ml, even into the area of sub nanograms per ml. This is our most sensitive assay. This protein is present around like 500 picograms per ml. And actually, with the newest, all what we have done so far is using mainly the adjuvant 6490. Now we have the 95, and then with 95, which is a little bit more sensitive. By factor 2 to 5, we can go even deeper into the proteome. But the cost over here is, of course, time. Instead of like running one 40-minute experiment, we have to do it 13 times more. And we're not only doing human plasma, also we have developed assays for urine, shown over here, very much the same. And here we can quantify around 130 proteins, all within one assay. And as you see, like we can generate reproducible curves for all of these proteins. And on average, we have a CV of less than 10% for these proteins. We have put out a bunch of papers which describes all these assays for human plasma, urine, CSF, saliva is coming, and also dry blood spots. And we are very excited about dry blood spots. I come to this a little bit later. But also now we have like a mouse. I mentioned around 100 proteins we can quantify in mouse plasma, and now we're going into the tissues. We have an assay already for around 160 proteins in heart tissue. And not only we are interested in heart, also like in lung, brain, and other tissues, which is what we're doing right now. And we hope that we have very soon assays for these different tissues, and from there, actually kits. So I mentioned that we are quite excited about human dried blood spots because of the reasons which are shown over here. Advantages: first, it's less invasive, and you can do it by yourself. You can poke in your finger by yourself with a needle. You don't have to go to the doctor, which is quite a if you want to go into population-wide screening. Then the initial processing is quite simple. Like you don't need to spin something down or have something on like the serum, like clocks it for a certain time. No, not at all. Just put the drop of blood onto a piece of paper and then put it into plastic bags, seal it, and send it off. So that's very important. There's no cold chain infrastructure involved, and it's like you don't have to freeze it, put it on dry ice, and send it by FedEx overnight to the laboratory. Now you can use just like regular snail mail, and which also saves a lot of money. And very important is like reduce biohazard risks, like HIV and other viruses. They won't survive after 20 minutes around on the dried blood spots. They're dead. So there are many advantages using dried blood spots. So the question is, can we quantify proteins using our MRM approach from dried blood spots? Here is shown the experimental overview. It's very much the same what we have seen before, but only like in the first step we are extracting, actually cutting out the entire spot here, and then we extracting the protein, and then we reduce the network and so on. And we can do this like the step by step this liquid handling systems. These are the proteins we are able to quantify right now. It's almost 100 proteins, 97 proteins. It's a list of the proteins, and you see many proteins which are of interest for a greater audience, like the Apolipoprotein, for example, the complement proteins, the complement factor proteins, and so on. And in this, there are around like many FDA-approved biomarkers as well. And it's actually like for many proteins the doctor is actually looking for. So on this slide, you see the expected ion chromatogram of the 97 proteins, and what you see over here is that you have very small peaks, and the reason is that we're using UPLC. A lot of people say like, well, if you're using UPLC and with bigger columns, then you're losing sensitivity and going to from a nanoflow to standard flow. That's actually correct, but we compensate for the loss of sensitivity by adding 10 times more on the column. And we can do this if we're talking about plasma. We have enough plasma. Even we have enough plasma for mouse. What finally goes on the column, even with our standard flow, is around 150 nanometers. Nanometers, not micrometers, 150 nanometers on the column. So we definitely have enough. And we switched like a couple of years ago from nanoflow to standard flow because it's much more robust. And the retention time is highly reproducible. We are talking to come out all the time on the second. So we are quite excited about this technology. And it gives you sharp peaks, as you can see. And you also see like for each analyte you have two peaks, one in red and one in blue. The blue corresponds to endogenous and the red to the cis peptide. And here, like we cannot quite cover 6 orders of magnitude, but 5. In our first highly multiplexed assay from dried blood spots. So in many of these proteins are represented not only by one peptide, by more than one peptide. And it's also quite important we're moving to more than one peptide per protein. Then the question is, how precise is this? Is it as precise as we have shown, we and others, what you can do using plasma? And indeed, it is. As you can see over here, we analyzed 6 samples and we looked for the intra and inter assay precision, and you see most of them, they are better than 10%. Now I have like here a movie which can show you how we actually get the exact amount of blood onto the blood spot. Because what we do, we cut out the entire spot. But so since we want to, in the end, we want to know what is the concentration. If we punch out, then as you know, like some blood spots, they spread out in a different way. But if we cut out everything, then we don't have this problem anymore. But we need to know exactly the amount of the volume of blood we are applying to the blood spot. And there are some very inexpensive devices. This one is one of them. It's called MicroSafe type blood spot collection device, shown over here. And I show you a movie. I hope there's some sound in the background. I'm sorry for this, but turn down the volume. Starting it. You see it like it sucks it up. It's very simple to use. It's out of plastic. I would also like so you don't get hurt. Then it stops right here. These are exactly 15 microliters, and then you apply it to your filter paper. And that's pretty much it. Then the question next is another device which is cheaper, it costs in 16 cents. And this has, but it has a glass capillary. It might be that people can get hurt with the glass capillary. You need to be a little bit careful. But so we compared this with the golden standard, which is of course a pipette. We were very surprised that we're getting very good CVs, and they're all around like 5%, are very comparable. So this is like really, really fantastic. And then the major question is like a lot of people say like, wow, dry blood spot is awesome. You can keep the protein for a long time. And we check it out. Like, we start the filter paper at minus 24 degrees room temperature and 37 degrees over a period of almost like a half year. And then we quantified the 100 proteins or around 170 peptides. And some of these peptides, they degrade right away, but most of these peptides, as you can see, like they're quite stable even over a long period of time. But it really depends on peptide to peptide, and you need to check it out. But these results makes us very, very happy. It shows like certainly like if you do this, you put it on a piece of paper, in most of the cases, like at least like within 7 days, if the blood spot comes to your lab in 7 days, and every male should come within 7 days, you are able to quantify most of these proteins. And also what you see is like there's not much difference between the different storage temperature, like so that's not the critical point, just like time. But again, like we are quite happy with this result. So now our what we want to do is like we want to make these assays available to the broader community by transferring these assays into kits. And the kits what we have developed are shown over here. We have the human plasma quality control kit to check out your instrumentations for the antigen 60.9 and 95, also for the size Q traps 4000, 5500, 6500, and as well for the thermal Q executive and TSQ Vantage. And now we also have brought in on the market the Biomarker assessment kit, which has 76 proteins in there, which you can quantify. And then very recently, since YUPO, we have Apolipoprotein kit and a quality control kit for mouse plasma and a mouse plasma bioassessment kit for around 81 proteins. So what comes with the kit? The kit should be like, ideally should be like waterproofed, and it really speeds up everything. Everything has to be provided, detailed protocol, and how to perform these analyses. And of course, it comes like with the internal standards. Everything is pre-prepared, that ideally less mistakes can be made, and it doesn't cost so much time and effort to perform these analyses. And in addition, It comes with all the LC-MS parameters, what you need to have to run these experiments for these different instruments. And then it also comes with a data analysis program we wrote by ourselves. So you get a license for this. It's only on our server. We constantly update our program, but I come to this a little bit later. And very important, it comes with reference or guideline values. Reference values, particular for the quality control kit. It means like if you run this, you need to get out this kind of result. So coming to the software, what we have written is called Qualisys, and this is a software program which automates the data analysis. It generates standard curve and assay performance determination will be performed, and then it applies the concentration of the samples to the curve and determine the concentration of the sample, and as well as comes with the quality assessment. As well as green, it's acceptable, yellow, take a closer look, and red, like we would not recommend to use data results. And this has been published in Journal of Proteome Research, I think like 2014, right? Yes, 50. And when this is an example, how it looks like if everything is fine, which is in green, it means like the data, they're looking here, this is our standard curve, they're looking, these are in green, that our data are in green, they're right on the curve, but if it's outside of the curve, out of the 95% confidence level, then it gives you, for example, a green light. Also it gives you a green light if it's like below the lower limit of quantitation or up to a limit of quantitation. So like immediately, like it's very important if you analyze a lot of samples, like you get like a immediate overview about quality of your analysis. This is like summarized like the proteins which are in the BIK76. It has 61 potential CBD markers and 30 potential cancer markers, and on like other markers which are somehow described in the literature to be connected to diseases. The dynamic range is at least like 10-fold for all of them, up to 500-fold. And then we looked at, so like, and a lot of people are asking that, so how comparable is this with the protein concentration you're determined to other methods, particular to the gold standard, which is ELISA. And for the BIK76, we found that 4 of them are being analyzed by the Mayo Clinic, and we could find the clinical reference range, and we could show that we analyzed some patient samples that they are within the reference range for a protein which is very highly abundant and a low abundance protein, as you can see over here. So it's quite comparable what we are doing, but sometimes, yes, the results are a little bit different what you have when you compare the protein concentration determined by ELISA versus LC-MS. This is our panel for apolipoprotein. And here, I mentioned this before, there's a lot of interest in apolipoproteins, particularly from the CVD, the cardiovascular disease researcher, and they're not so much interested in all of the other proteins. So like we just want to have, they're interested in the apolipoprotein, and so we developed an assay for them, and where we have 14 apolipoproteins, and what you can see over here, it can be done quite within less than 12 minutes compared to the biomarker assessment kit, which goes over 40 minutes. When we're talking about hundreds of samples, and really every minute counts, that you can make it shorter. Then we have a biomarker assessment kit for mouse and the quality control kit over here, and as you see, like most of the quality control kit, the idea is here like it comes with reference. So we check it out in our lab over here, and determine the concentration using different machines, and then this is like our reference value. And if you can determine the same concentration, then you can be quite sure that everything is okay. And we have actually sent out this quality control kit within the HUPO initiative for standardization to 16 labs around the world and ask them, like, can you analyze this? Then these 16 labs had, I think, 7 different LC-MS setups, and what we figured out is, like, all the instrumentation are all fine. It doesn't matter if it's really from Agilent or Sykes or even Bruker or Thermo, they're all doing a great job. It's not really the instrument. It's mainly like it's based on human errors, and which is demonstrated here. Like, this is like, we sent this out, like, this came back from an Australian group, and they determined that they have here like the standard curve, and you see the precision is quite good, and the R square is fantastic if we correlate this to, and then if you look at the concentration, what they determined for this protein, gasoline is 7.6, compare what we determined is 7.2. So that worked out quite well. But not so good for this participant over here. As you see, like, the precision is not really there, and this is mainly based on pipetting errors. And also they made another error. It's like they switched the vials. This vial over here should be there, and this one over here should be there. And these simple mistakes lead to errors, and which then have, like, which resulting, like, into something what you cannot use anymore. Like, but again, like, it's not on the machines. It's mainly the human mistakes which lead to these kind of results. But this can be checked using this QC kit. And also, like, you can check the performance of your instrument, like, over time. Here, for example, we performed the quality control kit on a daily basis on our one of our 6490s, and you see that what we recognize that the sensitivity all of a sudden is going down. This is all, like, all 35 heavy label peptides what we are detecting, and this is the average of all of them. But it's important that you look not only for one peptide, for ideally like for all of them. And you see, like, it came down over here, and then we really had to call in. We lost 60% of sensitivity. We had to call in a service engineer, and he checked it, and then said, like, no, it's ready to go. But it wasn't really ready to go. Like, it took us another day until the vacuum was stabilized, and we finally got back our sensitivity. You can do this. You can track your lower limit of quantitation and plot this and see how your instrument is performing. And we do this routinely now with our instruments of what we have over here. And it should look like this. And if it looks like more like that, and then we definitely know that something is not quite right with our instrumentation. So where we're going from here, like I mentioned, like we have some off-the-shelf, some kits, but what we see is, like, for many of our customers, clients, and collaborators, so, like, it's great, like, yeah, I like your BAK kit, but I want to have in addition, like, these proteins, and I'm not so interested in those proteins which are in the kit. So, and what we move right now is more into more customized kits, like what we are providing for our customers. And for that, we developed a software which is called MRM Assay Designer, and which is based on the experimental data we have acquired from all our work and from all of the peptides which are in the peptide trackers. I mentioned around 2,500 peptides already in there, and we know exactly how the experiment should look like. So this is like the customer comes into this web page and types in the protein of his interest, in this case apolipoprotein, and we have listed here on 16 apolipoproteins. And then it's kind of like what you know, like when you go shopping, like you click on here, like add this to my assets, and you can also view what do we have, what is known about this peptide, everything, all the informations are here. But what we want to achieve over here, that the customer is building up his own individualized assay. So, and then the next step is we provide them based on our experimental data, so we know exactly what the retention time is and all that. We provide them with a suggested MRM assay. And it's like, and we give them some options, like, do you want to have a very complete assay, with the most comprehensive assay, means like with all the peptides we have for this protein, or do you want to have, like, since you want to analyze thousands of proteins, you want to have the fastest assay possible. And we will build to them and come up with a design. And so this is what we envision how it will happen in the future. That's you, is a pretty looking woman over here, is typing in, looks like what proteins of her interest do we have in our peptide tracker. If we have some all there, then we design the assay, and then it comes back to her. And it's like, okay, this is our design. If you are happy with it, we're providing you with the peptides, and then, but then we also help you to optimize it if there are some questions. But we're not doing this in our own shop over here. It's more in collaboration with our client and customer. What if in the case if the protein is not there? Then we're looking at our picker library, and here we have analyzed the entire human proteome, as well our entire mouse proteome. And in the human proteome, for example, we found around 160,000 suitable MRM peptides. And if they're in there, but it's not in our tracker, it means that we haven't synthesized, we can synthesize this for you, and then we optimize in the assay and goes back into the tracker. And from there it goes in over here. What is in the case for the 160,000 peptides we have in here, that correspond to around 16,000 proteins. But what is in the case if you are interested in the proteins which are not in there? Then we can go back to our Peptide Picker and collect the little criteria. As I mentioned before, like we wanted to have like a not too short and not too long peptides between 7 and 20 amino acid residues. We can extend it. So like we go to 25. And see, like, can we then find a peptide which is then suitable for MRM? And if so, then we synthesize in this case to optimization again, and goes in the tracker, and from there we're building the design and goes back to you. So this is what we have in our mind. And in addition, we call it MedPipe, this pipeline. In addition, we provide statistical analysis. Like what is very important is outlier removal. I can give you an example where we have a client. We analyzed around 200 samples, and they looked at it like what the span of the concentration range of these proteins, and they came back to us like, so like this cannot be right. Like this protein has a different concentration or a factor of 1,000. That's not biology. That's something not quite right. And indeed, like then we did a statistical analysis. Like when we look at all of these proteins, there was like 190 proteins in 200 samples of the quantified. Looked at all of these samples, and there was really one sample out of the 200 method it up. Like when we removed the outliers, and then all of a sudden we found a normal distribution. And also this is very important, that we meanwhile, since we have analyzed thousands of samples, and we know pretty much where we can expect and what concentration we can expect the different proteins. We have these reference ranges for each protein, and we look it up. Like, and if it is within the range, you can be quite confident everything is fine. But if it's without it, then you might have a deeper look into your data, or it might be in biology, which is also quite interesting. And then, of course, like we track everything over time. So I think it's now I'm coming here to my summaries. It's almost like the hour is almost gone. We have developed many assays, MRM-based assays for quantifying human and mouse proteins in biological samples and biospecimens. We have rigorously assessed and developed these assays and rigorously assessed them. And many proteins in these assays, they are putative biomarkers for many diseases. And now we have transformed these assays into kits, not all of them, but we're trying to do this for as many as possible. We have the quality control performance kits for your instrumentation, and our first biomarker assessment kits, and also some specialized kits like the Apolipoprotein kits, and not only for human, but also for mouse. And we have developed a lot of software tools which can be of great value for the broader community and helps to develop new methods and to help in the data analysis and interpretation. So these are the names of the programs we have developed, and some of them are already published, and some of them will publish very soon. So I would like to acknowledge our people that worked on the assay development. It's mainly Andrew Percy here from our Proteomics BC Proteomics Center. Andrew Chambers was the leading scientist on the dry blood spot project, and Jun Thong is our main technician. And I would like to thank our bioinformatics person, Yassine, who developed all the different software packages, and our operation manager, Derek Smith. And then from here up, when we have developed these assays, then it goes to MRM Proteomics, where we transform them into these kits. And these are the employees, and of course, Gary Cooper, who is running running the company. And I would like to thank Agilent Technologies. We have done most of these peptides in collaboration with Agilent, and I particularly would like to thank Christy Miller and Steve Fisher. And actually, I think we forgot our funding agencies, which are shown over here, Genome BC, and I would like to thank you for your attention.
Thank you very much, Christoph. That was an excellent presentation. And you stayed on time, which is even more excellent. So we have about 5 minutes for question and answer. I will ask the first question, and if anyone from the audience has questions, please type them into the chat, and I will ask them after the first one. So I was wondering, for proteins that have more than one peptide, are the results from the peptide averages used to obtain the protein concentration, or how is that done?
So we have this quite a lot. Like when you have two peptides on the same protein, you not necessarily get out the same qualitative results, the same concentration. Then we have two proteins. Ideally, like when we go from 2 proteins to 3 proteins, and hopefully 2 of them they match. But first of all, like what is important is like that these two, the data, they need to correlate quite well. If they do, it gives us a lot of confidence that the quantitation what we have performed is in good order. And then in most of the cases, what we see is like when we look at the peptides, some of these peptides have potential chemotropic cleavage site in there, and these peptides are, in many cases, the peptides are giving us the lower quantitative result. And then in most of the cases, we take like this into our account and present the results from the peptide, which provide us the higher values. So not necessarily we averaging that, like we're looking more into this, like and then decide what's going on.
Great, thank you, Christoph. Okay, we have a question from the audience. what is the sensitivity difference between UHPLC regular flow and nanoflow? And then kind of following up on that, are there differences in ionization and ionization suppression? And can your assay be adapted for nanoflow?
So, yes, certainly it can be adapted for nanoflow. And then the first question was what's the difference between UPLC, regular flow, and nanoflow. We have checked it out, and we have published it. We checked it out with 12 different peptides, we have, no, there's many more peptides. It was 80, it's almost like 100 peptides. And we have shown in this publication that in most of the cases, when you're adding 10 times more on your regular flow is like microliters per minute, or like 200 microliters per minute. If you're adding, and you can use the 2.1 millimeter column, you're adding 10 times more on the column, and at least like you can achieve the same kind of sensitivity with nanoflow. In many cases, actually, you are more sensitive than nanoflow. So this was a study we have done a couple of years ago, and we have published it. And it has been confirmed by other scientists, other groups, that this is really the case. But of course, not for every peptide. And but if you don't have enough material, for example, you just have even biopsy samples or whatever, where you're quite limited, then we're going back to nanoflow. So certainly, yes, it is adaptable. When you have something developed for microflow, and then you still can go back to nanoflow.
All right, excellent. Thank you very much, Christoph. I think we've run out of time here. We are always available for calls, questions, or product information, so please feel free to contact either myself or another representative at Cambridge Isotope Labs. With that, we will end the presentation, and I wish everyone a great afternoon, morning, evening, and take care. Thank you.
Thank you.